Welcome back. Now we can continue our discussion of primary structure and protein purification. In this recorded lecture, we are going to focus on protein purification, and you will see how the knowledge of primary structure, or simply the amino acid sequence of your protein, can significantly aid in the decision to apply certain methods of protein purification versus others. Before starting any protein purification protocol, the first thing that you have to stop and ask yourself is, how will we detect the protein that we are interested in? How do we know that the application of a specific type of protein purification, a particular method of purification, is going to retain the protein that I am interested in, as opposed to losing it, and then you have nothing really to study on the back end when you're done? So the question here is, how will we detect a protein of interest? And generally, there are multiple ways to go about this, but we're going to focus on two of them. The first is going to be that of enzymatic activity. If your protein is an enzyme, it has some sort of catalytic function, you should, with each subsequent purification step, be able to follow the enzymatic function of that enzyme. You should be able to follow the reaction that it catalyzes, and that will tell you whether or not you have retained your protein. Okay, so if you think about this, it should make sense. The rate of product formation is going to be proportional to how much enzyme you have present. If you have more enzyme present, you should see that the reaction that you're interested in occurs with a higher rate. You have more catalyst, so therefore you should form product more quickly. So there are two general ways that you can go about this. You can do what's called a coupled detection. So on the left-hand side here, you'll see an example of a coupled detection. And something to make note of here, for both of the examples I'm going to give you, we are looking at an enzyme that catalyzes ATP hydrolysis. So you have two routes to look at the same thing here. Now, in the coupled path, what you have is an enzyme that basically takes one of the products of your ATPase reaction and uses that to do something else. Now, to be a little bit more specific, for an enzyme that catalyzes ATP hydrolysis, this most often means that you are converting ATP to ADP, adenosine diphosphate, and separately inorganic phosphate. So your products are ADP and phosphate. Now in the coupled assay, what's going to happen is, effectively, once your reaction is done, you are going to intentionally deplete all of your remaining ATP. You're going to basically remove it from the equation. And then you're going to regenerate from ADP a pool of ATP that can be used in the reaction that is shown here. Now, before I give you this reaction, I want to really highlight that this is not meant, I'm not expecting you to be an expert on this. I want you to sort of see that what we are doing is taking a product of your ATPase reaction and using it in a secondary reaction. Here, this is catalyzed by the enzyme firefly luciferase. This is the enzyme that gives you the glow that you see in fireflies. And this reaction involves luciferin, ATP, and oxygen, produces light, and the light that is produced can be used here to quantify how much ATP you have. Now, this is important because this can be done in different ways. This particular reaction can be done in different ways. In some paths, what people do is actually deplete the remaining ATP from your primary reaction, and they do so by converting all of the ATP to, say, AMP, or adenosine monophosphate. Now, what that leaves in solution at the end of your reaction is ADP that was generated from your enzyme. So in that case, what you can do, there are separate enzymes that you can add in that will convert ADP back to ATP so that you can use the ATP in this reaction and quantify how much ATP is remaining based on light production by firefly luciferase. So the only ATP that is there would lead to light. And so you can quantify how much ATP is there, which for every one ATP you would have one ADP that was produced. Now I know that this is all probably seeming convoluted. It seems like I'm giving you a lot of technical information. What I want you to see here is that basically we do our reaction and we use another enzyme to detect the product of the primary reaction that you're interested in. So what this implies is that there is not a way to directly follow the product formation of, say, ADP or phosphate. That's kind of difficult. Now, alternatively, you can do direct detection of products. So in the ATPase reaction, phosphate can directly react with certain metals to give color changes. This image is a very good example. You should see no phosphate, some phosphate, a little bit more, a little bit more, a little bit more. So you go from yellow to like this emerald green. And so based on the absorbance of the solution, you can directly quantify how much phosphate is present. So here you have two different paths that sort of give you the same information. You can quantify how much ATP is present in solution based on the activity, the enzymatic activity of a secondary enzyme, hence the coupled enzymatic reaction, or you can just directly detect product via reaction with something else, like a metal complex. These may seem like the same thing, and I can see how that would come across. But the point of this slide is really to just get you to realize that what we are doing, whether it be indirect or direct, we are following the enzymatic activity. So if the rate of product formation is proportional to how much enzyme you have present, having a method to observe, to quantify how much product is formed, is crucial to knowing how much enzyme you have present also. You have your assay, you have your method to observe enzymatic function. Now, what are you going to do with that information? Well, what we're going to do in the context of a protein purification protocol is apply something called the specific activity. We're going to use specific activity to quantify purification really efficiency. Are we actually, by applying a certain purification protocol, are we actually removing other stuff while retaining our protein of interest? Okay, so the specific activity is simply a mathematical way to express the activity of your enzyme of interest per milligram of total protein. So there are SI units that describe this, but generally speaking, the specific activity is the total amount of enzymatic activity, and this is activity that is observed using your established experimental method, and then that over the total amount of protein. So these are two measurements, but what it tells us is how much activity that would be associated with your protein of interest is there per mass unit of all protein. Now, if you want to think about this a different way, let's look at the example here. So I've got two flasks shown for you. Now, initially what you're going to have is a crude mixture. You have your protein of interest living inside of a cell. Now remember, this environment is complex. There are soluble metabolites, there are soluble proteins, there are insoluble lipid materials, nucleic acids, all kinds of things like that. So once you lyse your cell membranes and remove your insoluble material, you still have a crude mixture of stuff that just happens to all be soluble. So all of the marbles in this first flask you can think of as all of the different proteins that are present that are soluble in that crude lysate mixture. Now let's say we are interested in red marbles. The red marbles here represent the enzyme that we are interested in. So what we're going to do, if we know what the catalytic function of our enzyme is, and we have an assay, an experimental method in place to observe that function, we can observe the crude, basically the enzymatic activity of this crude solution. So we can measure the total activity, we can do that, quantify that, and that gives us this term here. And separately, we can use known methods to quantify how much protein is present. So there, we have a specific activity for our crude lysate mixture. Now then we're going to apply purification methods. And the goal of any purification protocol is to remove as much of your other stuff, remove all of your contaminants while retaining the protein that you are interested in. So basically what you're trying to do is maximize your protein relative to all of the other stuff in solution. Remember, I told you last time that the goal here is to have a solution of isolated protein that is at least 95% your protein, 5% other stuff. And really, that 5% other stuff, we want that to be as small as possible. So ideally, it would just be 100% your protein in aqueous conditions. The reality here is that usually you're going to be 95% pure, 98% pure. There's going to be a little bit of other stuff. So this flask on the right-hand side would represent a realistic scenario where you have a lot more red marbles than everything else, but you still have some other stuff present. However, specific activity allows us a way to quantify this. So you have your specific activity for your crude lysate mixture, and then what you do is you measure the enzymatic function of your solution after purification. So you can do this with each purification step or do it at the beginning and the end. But the core here is that you have to measure your enzymatic function, how much catalytic activity is there, and then how much total protein do I have. And so what you should see is that in a perfect world, the total activity would remain constant. So the total activity should be the same. You should have the same number of red marbles in the crude lysate mixture as you do in the final mixture. The only difference should be how much total protein do I have, because I'm trying to retain my protein of interest while getting rid of all of the other contaminating proteins that were initially present. So what this means is that my specific activity should go up. So I have a specific activity here in my crude lysate, but because I have reduced the total amount of protein in my final solution, this specific activity term should go up. The total amount of protein goes down, total activity remains constant. The ratio of those two is higher for the purified solution relative to the crude lysate mixture. Now this is important because it allows us a way to quantify the purification progress. Now, one thing to realize that what I just told you was an ideal situation. With any sort of purification protocol, the realistic picture is that you're probably going to lose some of your target protein along the way. With each additional purification step, you're probably going to actually have some loss. So this is not a perfect approach, but it still works. Now, a secondary measure here is percent yield. If you want to know what the percent yield is on each purification step, you can actually use your total activities here as a way to quantify that. If you have the total activity of your initial extract, the crude lysate that I was talking about, if you take the ratio of that and the total activity measured at any specific purification step, you can get some measure of percent yield. So how much of my protein do I retain after purification relative to when I started? Together, specific activity and percent yield are very useful properties, very useful parameters to be able to calculate to really quantify the progress of a purification protocol. You don't see tables quite like this anymore, but once upon a time it was very standard to report purification protocols in this way. So the table basically tells you everything you need to know. What step is being applied, how much protein is present, total activity, specific activity, and then you could use all of that to calculate, purification progress, as whether it be by percent yield or fold purification. And what I want you to be able to do is look at a table like this and at least to some extent interpret it. What I want you to see is that, as expected, the amount of total protein goes down from your crude lysate, here referred to as your homogenate, down to the purified protein. So I went from having about 3 grams of protein, total protein, my protein that I'm interested in, as well as all of my contaminant proteins, down to about 1 milligram of protein. So that is a very significant change in the amount of protein that I have present. But one thing to point out here is that my activity has changed also. And this is more of a realistic view of any sort of protein purification protocol. I'm going to start out with a defined amount of enzymatic activity. And perhaps the reason that I have lost activity is maybe there are multiple enzymes in the cell that catalyze the same reaction. I have a way to separate my protein from those other ones, and so I lose activity because of that. That is one explanation. But regardless, whether it be loss of material or loss of contaminating proteins with the same activity, I go from 27,000 roughly activity units down to about anywhere from 4,000 to 2,000. I'm trying to not complicate this too much, but I want you to see that you have crude mixture, first purification step, then second purification step, third purification step, and then the last purification step here. I can explain why there are 2 peaks shown, but don't dwell too much on that. Regardless, what you have is a change in the specific activity. The amount of total protein is changing, but so is to some extent my total activity. I start with a specific activity of about 10, and I go down to something in the thousands. So my specific activity goes up significantly, and this is really what you want to see. You want to maximize the amount of activity for the protein that you are interested in while removing all of your contaminant proteins. And you'll see that the purification fold is significant as well. I go as high as 350-fold increase in purification. Though my percent yield is low, I go from 100% down to 8 to 15% yield, but that is okay if I have retained as much of my protein as possible. Perhaps I have achieved my 95% or greater standard of purification. That's okay. As long as you have enough of your protein to do the experiments that you want to do, and that protein is pure, then that's really what you want. Okay, so at this point, we have the means to look at enzymatic function. We have a way to quantify catalytic function. But what I haven't really talked about is how do you determine the total amount of protein? Well, we can do this by spectroscopy. And you have seen this at some point, but we're going to spend some time here talking about the Beer-Lambert law. I told you when we talked about the amino acids that your tryptophans, tyrosines, phenylalanines, these are all aromatic, and they are, for that reason, chromophores that absorb light. Now, we're going to go through the Beer-Lambert law, which you saw in general chemistry and maybe also in organic, and often students have long forgotten this. They have put it out of their minds. So I'm going to review it. And I like to always throw this joke out here that no, we're not about to talk about beer. We're going to talk about the Beer-Lambert law. So perhaps a little bit drier. Maybe you want to have a beer as we go through this recorded lecture. That's one way to go. So just know that we're about to briefly talk about the Beer-Lambert law. Now, before we talk about the quantification, let's talk about what it is that we are physically trying to do. Well, you're going to have a cuvette. So that's what is shown here. I'm no artist. This is a crude representation of a cuvette, but all this is a glass or quartz container that contains a solution of your protein, or really any chromophore that it is you're trying to look at the absorbance properties of. Now, I'm going to put it into a spectrometer, and this sounds fancy, but all this really is something that has a holder for my cuvette and a light bulb on one side. So this light bulb, this is a source of polychromatic light. So I'm shining light into my cuvette, and when I say my light bulb is a source of polychromatic light, I should be more correct and say that in between the light bulb and my cuvette is something called a monochromator that allows me to select for specific wavelengths of light. But the point here is the same. I am shining light into my cuvette, and the molecules that are present in the solution within the cuvette, if they absorb light, then that means the light coming out on the other side is going to be a little bit less intense than the light was going in. So you should see, if your molecules absorb light, that the light coming out on the other side is a little bit less intense. The light's a little bit dimmer than the light was going in. Now, if your molecule does not absorb any light whatsoever, then the intensity of light going in should be the same as it is going out, because there is no absorbance. Nothing happened on inside of the cuvette to change how much light comes out on the other side relative to what went in. So first we can quantify this using something called transmittance, and this really is what it sounds. How much light is transmitted from one side of the cuvette to the other. So you can see this quantified over here. It's just simply the ratio of the intensity of light going in versus that coming out. That's what you're looking at over here. And it is expressed as a function of percent. So we have basically percentage of light that is transmitted. If you have full 100% transmittance, this would suggest that you have very little actual absorbance. The light is not really interacting with your molecule in solution here. If you have very little light transmitted, then perhaps you have a lot of absorbance. Your molecules in the solution are absorbing a lot of the light that's being put in. The percent transmittance and absorbance are related logarithmically. So if you look at the equation here, you'll see that absorbance equals your transmittance, and this is actually percent transmittance divided by 100. So the fractional transmittance, if I take the minus log of that, I get my absorbance. Now the thing to take note of here is that these two terms are inversely related. So when I had no interaction of light with my molecule, I had 100% transmittance. All of the light that I put in on one side came out on the other side of the cuvette. Now, in that case, I would have zero absorbance. But on the other end, if my molecule is interacting with light, and let's say that my transmittance is actually zero, what that's going to give me is absorbance of 1. So these are linearly related, and you should see that as I increase the concentration of chromophore of light that is basically my species of molecule that is absorbing light, my absorbance is going to go up linearly with increasing chromophore concentration. This brings us to the Beer-Lambert law. Now, mathematically, this is just an extension of what we have already talked about. The absorbance is, if we skip over this middle part, which really relates transmittance to absorbance, the absorbance is equal to E times L times C. Okay, so what are these? What is E and L and C? What are these magical parameters that you're talking about here? Well, E is your extinction coefficient. This is actually meant to be a Greek epsilon, but here I'm just writing out E. Your extinction coefficient, all it is is the likelihood that a chromophore is going to absorb a photon of light at a specific wavelength. So what this means is that you can define an extinction coefficient for any wavelength. All it is a probability term. When we talked about the amino acids, I told you that tryptophan absorbs light really well at 280 nanometers. It has a high extinction coefficient at 280 nanometers. This also means that the probability of absorbing absorbing a photon of light at 280 nanometers by tryptophan, that probability is quite high. In contrast, phenylalanine does not absorb light very well at 280 nanometers. So it has a very low extinction coefficient at 280 nanometers. The likelihood of absorbing a photon of light at that wavelength is very low. So to restate, the extinction coefficient, all it is a probability term that tells you the likelihood of absorbing a photon of light at a very specific wavelength. So in the Beer-Lambert law, the next term that we're interested in is the path length, L. So L, this is a fixed parameter. All it really is a distance term. If you look at a cuvette, light enters the cuvette, has to travel some distance before leaving the cuvette. The distance that it has to travel within the cuvette, the distance is also known as the path length. This is the path length. Basically, the path length is the known distance that light has to travel from one side of the cuvette before exiting the other side of the cuvette. Now, the path length is a known parameter. When I buy cuvettes from various manufacturers, they tell me what the path lengths are. I know what it is I'm getting. So the path length here is a constant. If I buy a cuvette that has a longer path length, I should expect that my absorbance is going to go up. And remember here, this is a product. So if I increase any of these terms, the absorbance goes up as a consequence. So if I buy a long path length cuvette, I should expect my absorbance to go up as a consequence. If I buy a short path length cuvette, I should expect that the absorbance is going to go down as a consequence. Okay. Now the last term here is the concentration of your chromophore. If we're talking about proteins, this is the concentration of my protein in the condition, basically in the solution that we're looking at. The solution being the stuff that's inside the cuvette. Okay, so a couple of things to point out here. Again, this is a product, so if I have an increase in any of these terms, my absorbance goes up. But usually, I'm going to know my extinction coefficient if I know the protein sequence, if I know the amino acid sequence, the primary structure of my protein I'm interested in. I can usually calculate an extinction coefficient, so that's a constant. I know the path length of my cuvette. I bought it. I have the manufacturer specifics that come with that. So my path length is the constant also. I can measure my absorbance experimentally, so that really only leaves the concentration. I just simply have to solve for the concentration. And usually that's what I'm looking for. If I want to talk about specific activities, I need to know how much total protein I have, so I can use the Beer-Lambert law to solve for the concentration of protein, therefore allowing me then to convert it into mass units. I can take the concentration, I know the concentration of protein in a given volume of solution. I can then cancel out volume terms and I'm left with a mass term. I can calculate how much protein I have in that solution. Therefore, I can then calculate my specific activity. So one thing to note here, because this is a product, my absorbance is going to be linearly tied, or my absorbance is going to be linearly dependent on the concentration of chromophore in my solution. So if I increase the concentration of my chromophore, I should see a linear response in that my absorbance goes up also. Now when we are talking about proteins, remember tryptophan, tyrosine, phenylalanine. These are the chromophores that are going to be present in a protein. You don't have, of the 20 amino acids, you don't really have any other chromophoric options. And for that reason, we're most often going to actually look at the absorbance properties at 280 nanometers. So if you recall, tryptophan and tyrosine both absorb light very well at 280 nanometers. Phenylalanine, not so much. It absorbs closer to about 254, 250, 260 nanometers. So we tend to rely on tryptophan and tyrosine. What you're looking at here is a plot of the extinction coefficient as a function of wavelength. And I really want to impress upon you the extinction coefficient is simply a probability term. This is the probability of absorbing a photon of light at a specific wavelength. So what you should take note of is that phenylalanine absorbs light much better at about 254 nanometers than, say, anything beyond 280 nanometers. The extinction coefficient goes to near zero at these wavelengths. Tryptophan, on the other hand, absorbs really well at 280, and it drops off as you go to longer wavelengths. So remember, this drop-off, because we're talking about extinction coefficient, we're talking about a decrease in the probability of absorbing light at any of these wavelengths beyond about 280 nanometers. So this is a probability of absorbing light. So just really, really, really try to remember that absorbance, it is going to depend upon the wavelength of light that you are using, and there are certain wavelengths where you're going to absorb light better than others. There are physical reasons for this. If you look at the various different kinds of conjugated systems, it really is going to center on the nature of the conjugated system of pi electrons that are actually absorbing light. But we're not really trying to get into that here. What I want you to understand is that because proteins contain really three different kinds of chromophores in terms of the indole side chain of tryptophan, the phenol of tyrosine, and the benzyl group of phenylalanine, based on the protein sequence, you can calculate an extinction coefficient that accounts for how many tryptophans, how many tyrosines, how many phenylalanines you have, and then you can calculate your extinction coefficient. So this term, yet again, is a constant. I want you to realize that. But it is a constant that is going to depend upon the protein itself, the amino acid sequence of the protein itself. So proteins that are very different in primary structure are likely going to have very different extinction coefficients as well. To make this point to you, I've gone back to protparam because I want you to see that we can calculate extinction coefficients, and the extinction coefficient is going to depend on the protein sequence. So what I'm going to do is I'm just going to do a bunch of alanines, and then, oh, okay, here's a tryptophan. Let me compute that. All right, so 44 amino acids, I get my molecular weight. Let me go down to the extinction coefficient. Oh, okay, it's 5,500. If you recall, that is the extinction coefficient for one tryptophan, for tryptophan free in solution. Now if I go back and I add another tryptophan, now 5,500 times 2. Oh, cool, okay, so add another tryptophan. Do it again, 5,500 times 3. Now if I start dropping in, say, tyrosines, you're going to see an increase again. Okay, because I'm adding in chromophores. But if I start dropping in other things that are not chromophores, I'm just typing in random amino acid letters at this point. Oh, I threw in some phenylalanines. Let's see what happens. Oh, my number didn't change because phenylalanine does not absorb significantly at 280 nanometers. So the point in me walking through this with you is I want you to realize that the extinction coefficient for a protein is dependent upon the primary sequence. The amino acid sequence for a protein is going to dictate what the absorbance properties are for your protein. And because Most proteins are going to have different amino acid sequences. Yeah, sometimes you do see proteins that have sequences that are very closely related, but they're still going to be different. Because of that difference, you're going to see changes per protein in the calculated extinction coefficient. Now, that is not to say that you won't have two proteins that both have, say, 3 tryptophans and 3 tyrosines, and so just by happenstance they do have the same extinction coefficient. That's a possibility, but the sequence itself is likely going to be somewhat different if you look at the order of amino acids from one end to the other. At this point, we have talked about some ways, of course, we're not being exhaustive here, but some ways to look at total enzymatic function, whether it be for your crude lysate or a purified sample of protein. You have a need to develop an enzymatic assay that allows you to look at the catalytic function for your enzyme in solution. So that's how you would get your total activity. And we have just concluded our discussion of how you might go about quantifying the total amount of protein. So these two things together allow you to solve for the specific activity, and this is something that you can use to follow along each step in a protein purification protocol. Okay, so next time we're actually going to start talking about the specific methods of protein purification. Here I have been trying to set you up for the big picture. Okay, what would I use? What tools would I use to actually quantify whether or not a protein purification protocol is effective or not? Next time we're going to talk about the actual purification steps. What properties am I going to take advantage of in a protein to actually purify it and remove contaminants?